All cells require adenosine triphosphate, or ATP, to function. This compound serves as the fuel or energy to carry out all cellular functions. Because energy can be neither created nor destroyed, it takes energy to make energy. It's a very basic way to put it. The process, known as glycolysis, is a major process to create ATP, which needs existing ATP to begin with, a molecule of glucose, a whole mess of enzymes, and electron acceptors to allow for reconfiguration. A glucose molecule has a phosphate group added to it by a pre-existing ATP, which renders it to an ADP. With the help of an enzyme, the glucose has the phosphate attached and becomes a glucose 6-phosphate, then fructose 6-phosphate after rearrangement. A second ATP adds another phosphate group, turning the molecule into a fructose 1,6-biphosphate, with the 1 and 6 indicating which carbons possess the phosphate groups. The former ATP leaves as an ADP, and the single fructose 1,6-biphosphate is broken in half to produce two 3-carbon chains called dihydroxyacetone phosphate. Though broken into two separate compounds, both will proceed through an identical sequence, so I will only refer to one of the remainder of the process. The compound goes through another rearrangement, turning it into a glyceraldehyde 3-phosphate. Another phosphate group is added and a redox reaction occurs as an NAD molecule is reduced by accepting a hydrogen from the carbon chain. The molecule is now a 1,3-biphosphoglycerate. An ADP encounters this molecule, removing a phosphate and becoming an ATP. The carbon compound is now a 3-phosphoglycerate. Again, going through reconfiguration, the phosphate moves from the 3-carbon to the 2-carbon, becoming a 2-phosphoglycerate. At this point, 2 hydrogens are removed by oxygen, causing the carbon compound to become highly energetic and destabilizing the remaining phosphate, now phosphoenol pyruvate. A second ADP counters the compound, removing the phosphate and becoming another ATP. The form of glucose is now a 3-carbon chain of pyruvate. There's another important process used by prokaryotes that does not involve oxygen. This process is known as fermentation, which is actually an extension of glycolysis. Fermentation is due to being unable to use an electron transport chain or lacking one altogether. NADH is continually oxidized, so the NAD may constantly be available to act as an electron acceptor. Pyruvate might also serve as a final electron acceptor, producing lactic acid. Acetaldehyde that is produced early in the fermentation when CO2 is removed from the pyruvate may also serve as a final electron acceptor, resulting in ethanol synthesis.